Good morning. Today we have to discuss on cytokinesis. What is cytokinesis? How cytokinesis may take place in the plant cell and in the animal cell. How cytokinesis takes place in the plant cell and in the animal cell. Commonly, cytokinesis takes place after karyokinesis. Cytokinesis takes place after karyokinesis. Karyokinesis I have already discussed in the last video. Karyokinesis is a division of nucleus occurred by following different subphases: prophase, metaphase, anaphase and telophase. So telophase is not the end of cell division. Cytokinesis marks the end of cell division. So how cytokinesis takes place? After karyokinesis, that we have to study in detail. In detail we have to study. Cytokinesis, the word cytokinesis, cytokinesis. You should understand what is meant by cytokinesis. Division of cytoplasm. Division of cytoplasm. Division of cytoplasm after karyokinesis. Karyokinesis. Division of cytoplasm after karyokinesis by formation of either cell furrow or division of cytoplasm after karyokinesis, means after division of nucleus, by formation of either cell furrow or by formation of cell plate. Cell plate is known as cytokinesis. This is the definition of the cytokinesis. Cytokinesis marks the end of cell division. Cytokinesis marks the end of cell division. It means after cytokinesis, new or daughter cells are produced. New daughter cells may produced. So, in the cytokinesis, what happened? Cytoplasm undergo division by formation of a special structure in the plant cell and in the animal cell. Cytokinesis in the plant cell takes place by different method, and cytokinesis in the animal cell takes place by following another method. In the same pattern or in the same method, cytokinesis may not take place in the both animal cell and the plant cell. So how cytokinesis may occur in the plant cell and in the animal cell, we have to study in detail. First of all, we have to study cytokinesis in the plant cell. Plant cell cytokinesis. Cytokinesis. Here, division of cytoplasm after formation of karyokinesis or after karyokinesis by formation of cell plate. Division of cytoplasm by formation of cell plate is known as cytokinesis. Cytokinesis. In the plant cell, the cytokinesis takes place by formation of cell plate. So, in the plant cell, cytokinesis by cell plate method. Cell plate formation. Cell plate formation takes place during cytokinesis in the plant cell after karyokinesis. You should know what is meant by cytokinesis. You should know what is meant by karyokinesis. Karyon means nucleus. Kinesis means division. Here, nuclear division is karyokinesis and cytoplasm division is cytokinesis. During cytokinesis in the plant cell, cell plate formation takes place. Then what is the cell plate? You should know what is meant by cell plate. Cell plate is a special structure for cell plate is a special structure for in the metaphase. Suppose this is the plant cell having cell membrane, plant cell having cell membrane and cell wall. You know the cell wall. This is the cell wall. This type of cell undergo cytokinesis after formation of two nuclei. After formation of two nuclei. Suppose these two nuclei are formed after karyokinesis. Two nuclei are formed. So these two nuclei possess nucleus, nucleolus and chromatin material inside the nucleus. So This is the nucleus, one nucleus and another nucleus. Two nuclear formation takes place after karyokinesis in single cell. And in the plant cell, cell plate formation takes place. Plant cell plate formation takes place. So, what is meant by cell plate? Cell plate. Cell plate is a structure formed between the two daughter nuclei in the center of the cell with the help of vesicles and here cell plate formation takes place. After prophase in the metaphysic stage, cell plate is formed by the vesicles secreted by Golgi bodies. Secreted by Golgi bodies and spindle fibers or tubulin protein. That type of tubulin protein and tubulin fiber or spindle fibers, fragment of spindle fibers and vesicles released or produced by Golgi bodies togetherly form structures known as phragmoplast. Here, formation of phragmoplast takes place. Phragmoplast. Here, phragmoplast formation takes place. Phragmoplast. Phragmoplast. Phragmoplast formation takes place. Such type of phragmoplast is formed by vesicles produced by Golgi bodies and spindle tubulin protein or tubulin fibers produced from the spindle fiber during cytokinesis. Spindle fiber already formed during anaphase, during prophase. These spindle fibers undergo disappearing in the anaphase. Before anaphase, some of the spindle fiber involved in the formation of phragmoplast. Such type of phragmoplast are the structures developed by the fusion of vesicle and tubulin protein. Phragmoplast developed by fusion of vesicles and tubulin protein or spindle fiber fragments. So what is the role of phragmoplast? These are the number of phragmoplasts attached to each other. Phragmo means pieces. Plast means bodies, membrane bonded bodies. Plast means membrane bonded bodies. Phragmoplast. Here what happened? Plast means membrane bonded bodies. And the membrane bonded body is developed by fusion of vesicles and tubulin protein. So the pragmoplast is a structure in which material present help for the formation of cell wall. So pragmoplast contain materials for formation of cell wall. Here what happened? Number of pragmoplast as developed by fusion of vesicle and the pragmoplast joined to each other, and it developed actually in the center and grew towards the periphery. So development of pragmoplast and secretion of the vesicles with materials required for the formation of cell wall. Actually, starch contained in the pragmoplast, which is membrane bonded, and that help in the formation of cell wall. Such type of formation of cell plate in the center of the cell, that is known as centrifugal cell plate formation. Centrifugal cell plate formation. Here what happened? The cell plate grow towards the periphery, from the center to periphery. So cell plate formation takes place centrifugally from center to periphery. So such type of cell plate formation takes place and secretes the starch or cell wall material. Here cell wall material may secreted. Cell wall material may secreted and grow towards the periphery. Such type of structure is known as cell plate. So this type of structure is known as cell plate. And this cell plate formation takes place from the material secreted or released from the pragmoplast. So here cell plate formation takes place, leads to division of cytoplasm. Cell organelles along with the cytoplasm, cell organelles also distributed equally in the daughter cells. So such type of division of cytoplasm is known as cytokinesis in the plant cell by formation of cell plate. Question may ask, what is mean by pragmoplast? Pragmoplast is a membrane bonded structure developed by fusion of vesicle and tubulin protein. Here pragmoplast contain cell wall material. Cell wall formation takes place in the plant with the help, and that is known as cell plate. Cell plate grow towards the periphery and division of cytoplasm takes place. So cell plate formation takes place in the plant cell. Here what happened? Cell plate formation takes place during cytokinesis of plant cell. Now what happened in the animal cell during cytokinesis? Cytokinesis in the animal cell. Cytokinesis in the animal cell. Animal cell. Cytokinesis of animal cell. Animal cell. Formation of cell furrow takes place. Formation of cell furrow. Here, formation of cell furrow. What is meant by cell furrow? Cell furrow is a ring developed in the center of the divisible cell. Center of the divisible cell. With the help of a ring formed by actin protein. Suppose this is the animal cell. This animal cell undergo division. Animal cell undergo division by following karyokinesis and cytokinesis. At that time, animal cell undergo formation of cell furrow or such type of constrictions in the center of the cell. Here, cell furrow formation takes place. This is the cell furrow. This cell furrow, baga, cell furrow. This type of cell furrow is developed and grow towards the center. So cell furrow growth takes place from periphery to the center of the cell. So cell furrow formation takes place centripetally. Centripetally, cell furrow formation takes place. And as the cell furrow grow towards the center, such type of cell furrow formation takes place due to ring formed by actin protein. Actin protein, microfibrils. So here what happened? Microfibrils are involved in the formation of cell ring in the center. In the ring, in the center of the cell, a ring of the microfibril formation takes place. And that ring undergo contraction. And that ring developed a cell furrow in the animal cell. So when the furrow leads to grow in the center and the two daughter cell formation takes place. Suppose such type of cell furrow, suppose this type of cell furrow leads to formation of two daughter cells. This type of cell furrow grow and is it visible? Is it visible? Such type of cell furrow leads to formation of two daughter cells. So continuously cell furrow grow and form two daughter cells. Such type of two daughter cell formation takes place. Each daughter cell possesses nucleolus with in the nucleus, daughter nucleus. So such type of two daughter cell formation takes place. These are the daughter cells. Daughter cells. So these are the daughter cells formed by cell furrow formation after karyokinesis. These are the daughter cells. Daughter cells are formed by following karyokinesis and cytokinesis. First, karyokinesis takes place and cytokinesis followed by formation of two daughter cells. So cytokinesis means division of cytoplasm either by formation of cell plate or cell furrow. After karyokinesis, here most important thing that the cell plate formation takes place in the plant cell and cell furrow formation takes place in the animal cell. Now what happened if there is no cytokinesis? What will happen? If there is no cytokinesis, what will happen? The multinucleated cell formation takes place. Multinucleated cell formation takes place. Without cytokinesis, cytokinesis leads to the formation of multinucleated cell in the plant cell, that is known as plant cell undergo xenocytic condition, and in the animal cell when does not undergo cytokinesis leads to the formation of syncytium. Syncytium. Such type of syncytium means the cell division takes place in the animal without following cytokinesis. In the animal cell, multinucleated cell formation takes place without following cytokinesis and leads to the formation of the multinucleated cell. That multinucleated cell is known as syncytium. The term used in the animal cell, syncytium. And here in the plant cell or in the fungi, the term may use xenocytic multinucleated cell. And another phenomenon may observe during cytokinesis. Here what happened? In the plant cell, at some places what happened? The gap remain for the exchange of cytoplasm or protoplasm between the daughter cell or between the cell. That type of space remains without formation of cell wall. That type of place is known as plasmodesmata. Plasmodesma, singular, and plural plasmodesmata. Here plasmodesmata formation takes place. During cell plate formation, what happened? At some places space may be released and that space leads to the formation of plasmodesmata that help in the exchange of protoplasm between the daughter cell. So such type of cytokinesis may take place in the plant cell and in the animal cell.